potentials will be stimulated or initiated by a stimulus. They're going to be a small change in the membrane potential. The magnitude is going to vary. So what we will see for the action potential is that it's all or nothing. Each action potential is identical. There's no variation or gradation in the magnitude. For graded potentials, there is. So there can be a small graded potential, there can be a medium, there can be a large, right? That is not the case for action potentials. They're going to spread by electrotonic conduction, which simply means that it's going to be decremental as it moves away from the source of that stimulus. So it's going to be spread by the change in the membrane potential, but the magnitude is going to be smaller the further away we move from that initial stimulus, all right? It's going to be decremental or decreasing as it moves away. So here's the site where we recorded the stimulus. Notice how it's a large change, and by the time we record the membrane potential at the axon hillock, it's much smaller, right? Because it's decremental or dissipating. And then up here, we can see the variation. So we can have a small graded potential, and we can have a stronger stimulus, which gives us a slightly larger graded potential, et cetera, until we get to that -55. And then every single action potential is going to always be the same amplitude, the same magnitude, the same length of time, right? The same duration. Whereas graded potentials are going to be varying. The purpose of graded potentials is to determine whether or not it's enough of a membrane potential change to result in an action potential. So this is where we get that threshold concept from. It's the level of depolarization that's required in order to meet the criteria to generate an action potential. So the graded potentials can be excitatory if they are depolarizing changes. So depolarizing changes make the membrane potential more positive, right? So in other words, positive ions move into the cell. So the membrane potential goes from -70 to about -60 or so. And then if we have a hyperpolarizing change, this would be an inhibitory type of graded potential, and that would make the membrane potential more negative, so about -80 or so, and that would be further away from our resting membrane potential. So if we had a hyperpolarizing change or a hyperpolarizing, inhibitory graded potential, that would actually make it harder to fire an action potential, because that would make it harder to get to that -55 threshold. So depending on the type of graded potential that we see, we can either make it more favorable or easy to get to that -55, therefore easier to fire an action potential, or we can make it harder by making a hyperpolarizing change, which would put us further away from that -55 threshold requirement, okay? So think about the type of graded potential and its direction away from that resting value. The other piece here is that we can sum these graded potentials. We can have temporal summation, which is if you have the same stimulus repeated close together in time. So this is at the same spot on the neuron, right? So if we think about our neuron that we just drew, we had different synapses happening on the soma. And so if we took one of those synapses and we had two graded potentials come in, but they were so close together in time, we could just add those together, and those would potentially get us closer to -55, and we would then be able to generate an action potential. So temporal summation, same spot on the neuron, but they're just close enough in time. For spatial summation, this is if we have two different synapses. So if you think again about that neuron that we just drew, we had one synapse on one area of the soma, another one on another spot. Now, if they came in, or if graded potentials came in at the same exact time on different spots, we could sum those together as well. So spatial summation is if we have overlap graded potentials that are coming in at the same time, but they're at different synapses. Those could also be aggregated, and they may get us either closer or further depending on the specific type of graded potential. So here we can see we had a depolarizing graded potential at synapse W. We had a hyperpolarizing graded potential at synapse Y. And so we would sum those together. Now, because these were happening in opposite directions, like these were both sort of kind of balancing each other out, when we sum those together, we simply get back to rest, right? They just cancel each other out. If they were two depolarizing events, you would see something that looks more like this. Okay, let's look at a checkpoint question here just to test what we've said so far. Why are graded potentials unable to communicate over long distances in time? So think about this question and share your responses. So lecture objective 2, explain the ionic basis of an action potential, describe the gating mechanisms for voltage gated sodium and potassium channels. So what we're going to move on to describe here, and we're just going to introduce this and talk much more about it when we meet next time. But basically, what channels are open in order to begin generating the action potential? And so we just described that it's basically the voltage gated channels, which are concentrated axon hillock and then the axon. So we're going to talk about the gating mechanism of those voltage gated channels and how that results in the action potential. And more specifically, why that action potential can be sustained and travel for long distances. So the membrane potential changes that we see are basically the resting potential, which is our reference point, and that is -70 millivolts. Then we can talk about depolarization, which is going to be a positive change to the membrane potential. We can talk about repolarization, which is going to be resetting that depolarizing event, excuse me, resetting that depolarizing event back down to rest. That would be a repolarization. And then hyperpolarization is if we undershoot the resting membrane potential, and this is a more negative change in the resting or from the resting membrane potential. So a depolarizing event, a repolarizing event, and then a hyperpolarizing event, which would be much more negative. We also want to just reiterate, so we illustrated these channels, but I just want to reiterate where each of these channels are on the neuron and what they're responsible for. So leak channels are always open. They're going to be moving ions according to the concentration gradient, and they are going to be found throughout the neuron, and they're going to be really contributing to the resting membrane potential, as we described previously. Ligand-gated channels are going to be found on the dendrites and the cell body. They're going to be opened or closed depending on the ligand that's bound. And when we say ligand here, we're talking about a neurotransmitter or another ion, right? So these can be different types of ligands, but something has to bind to open up that channel. The voltage-gated channels, again, are open or closed depending on the membrane potential. So most of our voltage-gated channels would be closed when the membrane potential is -70, because that is the resting membrane potential. And we don't necessarily have any excitement happening, so that voltage-gated channel is closed. They would open up at that -55, and that's when we would fire the action potential. Now, voltage-gated channels are going to be sodium or potassium. They're going to be throughout the neuron, but more concentrated along the axon and even more so along the axon hillock, right? And they're going to be contributing to the action potential. Calcium channels are going to be found on the axon terminal, and they're going to trigger exocytosis, which is the mechanism for releasing neurotransmitters in those synaptic vesicles. And those can be dumped into the synapse. And just to kind of make the connection here, when the calcium channels open up and they trigger the release of neurotransmitters, these are the neurotransmitters that will go and bind to the ligand-gated channels and open up those ligand-gated channels. And this is how we can communicate a signal from the axon terminal of one neuron to the soma or cell body or dendrites of the adjacent neuron. So definitely make the connection between the neurotransmitters that are released at the axon terminal and these neurotransmitters acting as the ligand that will open up the ligand-gated channels that are located on the adjacent neuron, specifically targeted on the dendrites and in the cell body. All right. And then this is how we can really begin to see that neurons are speaking to each other, they're communicating to each other. Not only are they receiving inputs from presynaptic neurons, but the postsynaptic neurons are then sending those inputs to other neurons. So if we kind of zoom out and look at the big picture, the nervous system is really a highway of communication, right? Lots of neurons, and there are billions of neurons in our body and millions of synapses. So there's a lot of high volume and high speed of communication. And this is basically the language of that communication, the opening up of channels, the movement of ions, and then the generation of these signals. Okay. Let's talk about the ion gating mechanisms here a little bit closer. So changes in the membrane potential are controlled by changes in the flow of ions. This is why it's so important for you to think about the permeability being simply a reflection of what channels are open. When I talk about permeability increasing, I simply mean more channels for that ion open up, and so those ions can move more freely through the membrane. Potassium is going to have two types of channels. It's going to have the leak channels that we refer to as far as having that greater permeability. And then it's also going to have the voltage gated channels, excuse me, which are going to be important for stimulating the stimulating the action potential, right? Particularly repolarizing the cell so that it can get back down to the resting membrane potential. And we'll talk about that much more. Basically, potassium has two channels, the leak channels and the voltage gated channels. Sodium has many more voltage gated channels. And we know this because we talked about the fact that sodium's leak channels are very, very few. There's a very low permeability for sodium to leak across the membrane. So it has few leak channels and it has mainly those voltage gated channels. And the voltage gated channels are going to be important for opening up and then generating or stimulating the depolarizing phase of the action potential. Okay, and I want to begin to make the connection here. Because potassium wants to move out of the cell, that's going to allow positive charge to leave the cell. So when a voltage gated channel for potassium opens up, that's going to result in a repolarizing event. It's going to get us back down to -70. It's going to be a negative change in the membrane potential as that positive charge leaves the cell. Sodium, on the other hand, has an inward rush. So when I open up a voltage gated sodium channel, lots of that sodium, lots of that positive charge will rush into the cell, and that's going to be responsible for the upward spike, the depolarizing phase of the action potential. So as we think about the action potential and we think about which ion channels are opened up, I also want you to connect what we've said before about the overall ECDF for each of these ions and the direction of that ECDF. Because whenever the channel opens up, those ions are simply going to move in the direction of that overall electrochemical driving force. Okay, so definitely connect that to what we've said before as far as the movement of these ions. Okay, we'll look at the voltage gated sodium channels first and the mechanism for how these channels open up. And we'll talk about the specific gates and mechanisms on this channel. So the overall principle here is that the voltage gated sodium channel is going to open at that threshold value. This is why that value is so important, because when the membrane potential gets to -55, it triggers the opening up of these voltage gated channels. Again, that's the voltage mechanism or the voltage criteria that we're referring to in that name. This is called the threshold value. When these channels open up, sodium is going to rush into the cell. Again, think back to sodium's ECDF. Its chemical driving force into the cell It's electrical driving force into the cell. So it doesn't matter what type of channel we're speaking about. As long as a channel is open for sodium, the movement for sodium will always be the same. It's going to rush into the cell according to that ECDF. When that happens, the membrane potential is going to begin to climb. And again, think back to what we said the last time. Whenever I open up a sodium channel, sodium is going to try to move. It's going to try to approach its equilibrium potential. And we know that that is positive 60. So whenever I open up these voltage-gated sodium channels, sodium rushes in. The membrane potential will climb in an attempt to approach the equilibrium potential, which is way positive at positive 60. And that is going to be resulting in the depolarizing phase of the action potential. Now, because these channels, again, they're voltage gated, they're going to be deactivated at positive 30 millivolts. And so that means that sodium will never actually reach its equilibrium potential, which is positive 60, because the channel is actually going to be deactivated and closed at positive 30 millivolts.